everybody. Welcome and thank you all so much for joining us today. We're so excited about our September Ask the Experts webinar. We appreciate you coming in today. We are hearing about research projects from Dementia Alliance's Lina May Edwards Young Investigator Research Grant recipients. And we're just so thrilled to be able to do this with all of you. I'm Lisa Levine. I'm the Programs Director of Dementia Alliance, and thank you for joining us today. We really appreciate it. For those of you that are unfamiliar with Dementia Alliance, we are a North Carolina-based service-oriented nonprofit. We provide resources, education, and support to family members and people living with dementia. And we will put into the chat our information for you so you can find our website and join us and learn more about what we do. But research is a very important part of that. We have for a long time funded research. And in 2018, we established a scientific advisory board or a panel to better target our grant awards so that we can innovate therapies. These are seed grants, if you will, and hopefully they serve as a catalyst to help new and emerging dementia researchers and investigators with their research and also help them advance their careers. Our grants are awarded to researchers in North Carolina in multiple disciplines, and they are up to $100,000 for 1 or 2 years, and they are funded by private donations like donors like you, and also donors like our friend Laura Gaddis, who we have here today. Laura is the founder of Guardian Angel. And Laura, thank you so much for joining us. I know you're a board member here at Research is Really Close to Your Heart. Can you share with us the background and why you founded Guardian Angel?
Sure, Lisa, thank you. Good morning, everybody. I'm the founder and the president of Guardian Angel Thrift. My mother, Lina May Edwards, struggled with Alzheimer's for 12 years. Near the end of her life, I asked for guidance in creating funds to fight this disease. As I woke the next morning, I went to the kitchen table and wrote out the plan that would change my life forever. We raised funds for research grants through the sale of merchandise donated to our thrifts. I truly believe we have reached hundreds of thousands of people and raised awareness for the need of Alzheimer's research through our donors, our buyers, our families, and the general public. The 3 $100,000 grants discussed today were made possible through the Guardian Angel. Alzheimer's is a cruel disease to all who are exposed to it, the patient and the loving family and the caregivers. As an Alzheimer's patient starting down their rabbit hole near the end of their life, there's nothing that can reverse Alzheimer's at the present, only meds that ease and delay. That is the reason research is foremost in our mind. The Guardian Angel motto is Alzheimer's, research is the answer. Thank you very much. And I want to ask everybody to, when cleaning their closet, remember to bring us, that's how we get our money. Reselling merchandise. Uh-oh. I can't hear you, Lisa.
Sorry about that. Doing too many things at once. Thank you so much, Laura. We not only appreciate all that you do as a board member, but all the hard work that you do in raising funds. I know it's a very personal story for you, and we just can't thank you enough. Laura is one of our biggest supporters, and so go to Guardian Angel if you can. It's quite the experience, and it's a wonderful thrift store. So Laura, we're going to mute you. There we go. And I did want to remind everyone that research grants are funded primarily by private donations. So if you're interested in donating toward research, the best way to do that with Dementia Alliance is to reach out to our executive director, Heather Hooper, who's in our audience today. And she can have a conversation with you and you can learn more about how to make that happen, how to use that funding for research. So we will drop her email to you in the chat, or you can call our telephone number and have that conversation with Heather. So we all thank you very much. We are going to get right into the fun and juicy part now. that I'm so excited about. So we're going to start by talking to our researchers. We've asked that they share their projects and with you for about 15 minutes each. And then after everyone has presented, we will have a conversation, a discussion, and we will answer your questions. So while you're listening, if you would like to type any questions into your chat box, which is at the bottom of your screen or top, and it says chat, feel free to click on that and type in any questions that you have. We will gather those together and then we will try and answer those at the end of today. So first, we are going to talk with Dr. Jessica Ellis, and she's a PhD. Thanks again for being here today. You're an assistant professor at the Department of Physiology at ECU. Is that right? Great. And we know that Dr. Ellis, you received your PhD in nutritional biochemistry from UNC, that you're a postdoctorate fellow in the Department of Biological Chemistry at Johns Hopkins, but your current work is through the Ellis Lab. And I've been doing a little research and looking at some of your websites and stuff, and it looks really interesting. I can't wait to read more. The funded research project that Dr. Ellis had was deciphering the role of DHA in the development of brain disorders. And we are so excited to hear what you've got to share with us. Take it away, Dr. Ellis.
All right, well, thank you so much for the introduction. I'm really happy to be here today to share with you some of the work that my lab has been able to perform with the wonderful, tremendous support from the Dementia Alliance. Can you guys see my PowerPoint right now? You good?
Yes, we can see.
Okay, just wanted to make sure.
You sure can.
Okay, so our lab has been studying fat metabolism, basically in the brain. And so we're excited to share some of the results that we have generated over the last couple of years on this project and give you an idea of where it's going. So, you know, as Lisa mentioned, I have a background in nutritional biochemistry. So while we do a lot of biochemistry in my lab, we're always thinking about how it relates back to day-to-day habits, especially regarding, you know, the foods that you eat every day. And we have particular interest in the foods that we get energy from, the macronutrients, and those are broken up into three-ish categories. So carbs, protein, and fat. And so many decades ago, you know, the obesity epidemic began, and a lot of thought went out there to say, don't eat so much fats because we think that's what's making the obesity epidemic. So we stopped eating so many fats and turned more towards carbs. And with that, the obesity epidemic just continued to accelerate and diabetes became rampant. So then we revisited, well, maybe fats are not so bad as we originally thought. And so based on a lot of research, what's happening now is that we kind of are grouping fats into two different categories, the bad fat and the good fat. The bad fat is all that greasy, delicious fat that you have in your fried foods and other cheese-based foods. Traditionally speaking. And then the good fats are what you find in things like oils and nuts and some fatty vegetables, as well as seafood. And so we're going to kind of dive into the good fats today. And so when I talk about good fats, throughout the rest of the presentation, I'm really going to focus on what I'm going to call omega-3 fats. And these are the fats you find in fish. So if you eat fish as part of a healthy diet, or you do fish oil supplements or omega-3 supplements, it's likely that you're trying to get the really good fats that you find in these aquatic species. And so why do people take these fish oil supplements or just eat fish as a part of a healthy diet? And it's because there's gobs of research out there that have shown that it's really good for multiple aspects of our physiology of our body. So joint health, immune health, heart health, but also a lot of brain health benefits. So from varying different kinds of aspects of brain health, it seems to be pretty tremendously beneficial. And so, what would that have to do with the brain? So I just want to give you a little background on that. So fat in the brain actually makes up about 60% of the brain. So majority of the brain is fat. So you have your fat tissue in your body, which is the fattiest tissue that you have. But the second fattiest tissue in your body is actually your brain. So there's a lot of fat in this tissue. And here you can see all these arrows are pointing to different things that the omega-3 fats have been found to be beneficial towards helping. So why do we have so much fat in our brain? So there's many reasons, but here's a couple that I thought maybe we could all relate to. So neurons transmit information from within our brains and out to the rest of our body. And that information is transmitted sort of like in the form of an electrical current, like an electrical pulse. And so in the body of the neuron, you make an electrical pulse and it transmits all the way to the end of the cell. And then when it gets to the end of the cell, it gets released onto the next cell, which then does what it does with that information. And that's how information gets passed throughout our body. Now, as this electrical signal is going along the body of the neuron, it goes down this axon, and around that axon is a very thick layer of fat. And that is like a wire for electrical wire that has a coating around it. It's a very similar process. So the fat kind of keeps it insulated. It keeps the electrical current bound inside within the cell, so it's not going crazy and being released all over the place. And it also serves as a really good conduit. So it allows it to transmit pretty quickly. So fat's really important for that process. And then when the electrical current gets down to the end of the cell, it releases basically a whole bunch of little balls. And so these are your synaptic vesicles. And these balls contain your neurotransmitters predominantly, like dopamine, serotonin. But the balls on the outside, what they're made up of is fat. So those balls are made up of fat. And so without fat, we can't not only transmit this electrical signal very efficiently, we also can't transmit the information from one cell to the next cell without this fat. And it seems to be that the kind of fat that's in there makes a difference as to how well these processes occur. And the idea is that omega-3s kind of help facilitate a lot of these processes. And to get an idea of, okay, so what's so special about omega-3s? Okay, so let's just compare two different kinds of fats. Sort of the bad fat, which, it's you can still eat it in moderation, don't you worry. Just be sure to incorporate good fats into your diet as well. So the bad fats that we talk about are what's called saturated. So that means they're sort of like a straight line. And so they don't get to move around too much. And that makes them really rigid. And so that's what you see, you find a lot in hard cheeses, for example. They're literally rigid. And then these other fats that are the PUFA fats, the good fats, they have kinks in them. And the kinks allow them to be flexible so that they can move around a lot. And that flexibility, is like what you see in oils or in avocado, very squishy, right? So that seems to be good in order for your brain to function at its best capacity, sort of the way that we view it here. So surprisingly though, however, we actually don't understand very well how fat that you get from a fish, the omega-3s, goes from our digestive tract into our brain. And there's a lot of kind of mysteries around this. So for example, if you take individuals or animal models, that have increasing levels of omega-3s in their blood, like you can measure omega-3 index in the blood, what you find is that in the muscle tissues, the liver, tissues like that, it goes up correlationally, right, as you go up. But in the brain, it kind of remains flatlined. So it seems that while the brain has a higher overall content, you can see that it's hard to manipulate the levels of omega-3s in the brain. And we don't really have a good handle of this on humans because we can't take a brain sample and figure it out. So we've done a lot of this research in animal models. And so trying to figure out what it is that allows the omega-3s to get into the brain and to where they need to go. And so that has been the main question that we've been asking in our lab. And we had a hypothesis that there was a particular protein that was taking these omega-3 fats from the blood, and once it gets into the brain, was metabolizing it so that it could go into the membranes of our brain, which is where we want it to be in order for it to serve its best function. And so this is the protein we targeted. I'm not going to keep mentioning it or go too deep into it, but in order to determine if our hypothesis was correct, what we did was we knocked it out in an animal model. So we knocked it out in mice. And then the idea was that we were going to say, okay, so if we get rid of this, does this cause less omega-3s in the brain? And then what are the consequences of that? So we knocked it out. And of course, the first thing we did was check the omega-3s. And we found that there was indeed a 50% reduction on omega-3s in the brain of these animals. So what you're looking at here is sort of omega-3 map in the brains. Here's the control brains and here's our knockout brains. And so you can see there's a pretty like intense green, red signal in our controls for the omega-3s across the brain. In our knockouts, the signal is just much depleted. So these brains are just lacking omega-3s in their membranes pretty much across all of the regions of the brain, which was remarkable. We were very excited our hypothesis was correct. And this is like a one-in-a-kind model. There really isn't anything else out there like this at all. And so we were really excited, and we have been wanting to use our knockout model to study omega-3s and their role in brain health. So we started by looking at its impact on aging, just the natural progression of aging. And we're pretty much done a lot of those studies. And now we're moving on into its impact on age-related diseases. So targeting Alzheimer's disease as well as Parkinson's disease. And so results from our aging study is essentially that they don't age very well. So you don't have to know exactly what this means. I'll just tell you that what we see here from these images is that our knockout mice have indicators that suggest that they have injury in their brain, but they did not undergo any kind of injury. All that they did was be allowed to age. And so having less omega-3s in their brain caused them to look like they had an injury in their brain. So obviously omega-3s are protective across the aging spectrum. And we also found that a lot of the proteins that were involved in this synaptic process, the transmission of information from one neuron to the next, were all dysregulated. And this got worse with aging. And so we conclude that these neurons are just not really communicating properly. And so this indicates, omega-3s are obviously good for aging health. This manifests in numerous ways, but for example, they have impairments in their memory. The knockout animals have poor short-term memory, which is suggestive that they might be predisposed to something like a dementia. And they also have hyperactivity. So they move around more, they move faster, they stop less. So this was indicative to us of sort of maybe on the lines of increased propensity for Parkinson's-like disease. So to begin our disease model investigations, we have started in the lab a project related to Parkinson's disease. And for this, we used an animal model that has Parkinson's disease. And this is a common model to use in research for Parkinson's. And this is an image showing you alpha-synuclein. The red signal is alpha-synuclein, which is a protein that is known to accumulate and be part of the progression, the disease progression for Parkinson's. And so these are our controls, and this is the Parkinson's disease model. So you can see that there's a little more to signal in the Parkinson's disease model. And then when we take the Parkinson's model and we cross it with our knockouts, so now we have Parkinson's with 50% less omega-3s in their brain, this is what we see. We can see a huge increase in these alpha-synuclein aggregates, suggesting that omega-3s are very important for the disease. Much more strikingly, however, this Parkinson's model doesn't live quite as long as the average mouse. They succumb to the disease of the disease model. And when we cross these mice to our knockout mice, they actually don't survive very long at all. They have a much a poorer survival rate compared to even the standard Parkinson's model. And this is very, very striking. It's more striking than anything we've seen in the research literature. So we think this is excellent evidence that omega-3s in the brain are extremely important for the onset, potentially, and progression of Parkinson's disease-like symptoms. And then related to Alzheimer's disease, I don't have data to share with you. However, I have been working with a collaborator at Columbia University, and we actually recently acquired NIH level funding, a really great grant in order to study Alzheimer's disease in our model. And so we're going to do a similar type thing. We're going to take animal models of Alzheimer's disease like this one, where you can see here the control has a lot of omega-3s in its brain, and the Alzheimer's model is depleted of these. And we're going to ask the question whether, depleting it, the animal of omega-3s before the onset of the Alzheimer's disease progression makes things worse. And we're going to try to understand the mechanisms there. So we have about 5 years worth of funding through this mechanism, and we're getting underway. It's starting 2 months from now. And so we're going to get underway to understand more about the link between omegas, the protein that we have targeted, and the progression of Alzheimer's disease. So with that, I just wanted to thank my lab, especially Regina Fernandez here. She's my doctoral student who did almost all of this work. She just graduated with her PhD and is now doing a postdoc. And my master's student, Amber, is taking over the project. And I will say that the Dementia Alliance was critical in providing the support for me to fund my students so that they could do all this amazing research. And so I'm very grateful. With that, I will turn it over to the next speaker, and I look forward to your questions at the end of the panel.
Thank you so much, Dr. Ellis. That was really interesting. I know we're going to have a lot of questions come up. I've already seen some. So give us a few minutes and we will definitely send those over to you. Exciting research there. Okay, we're going to move on now to Dr. Daniel Parker. And Dr. Parker received his medical degree at and had an advanced fellowship and did internal medicine residency at Eastern Virginia Medical School. And his postdoctoral research fellowship was with the Duke Center for Study of Aging and Human Development at Duke University School of Medicine. He is currently an assistant professor of medicine at Duke University and a practicing geriatric medicine specialist and internal medicine doctor with Duke Health. And his research project was titled The Effect of Exercise Mode, Amount, and Intensity on Plasma Markers of Brain Aging in Healthy Adults. And we are so happy to have you, Dr. Parker. If you want to go ahead and jump in there.
Thank you so much for that introduction. Can you see my slides?
Yes, we can.
Okay, wonderful. So first off, I just want to thank Dementia Alliance of North Carolina and all the donors who made these wonderful contributions that have supported this line of work. And so just a little bit of background about me. So I'm a geriatrician, memory disorder specialist, and physician scientist. And my clinical work is at the Duke Memory Disorders Clinic, where I evaluate patients with cognitive changes and diagnose and manage dementia and cognitive impairment. And my research is really focused on developing therapies to prevent dementia. And the approach I'm taking to do that is looking at interventions that we know improve brain health, like exercise and healthy diet, and trying to understand what's going on at a molecular level that leads to improvements in brain health, and then trying to identify drugs that we can use to recreate those effects. And so I'm often asked how I got interested in dementia and geriatrics. So on the right here, you see a picture of the forest at Duke. So this is a continuing care retirement community in Durham. My mom worked there. And when I was in middle school, I would go with her to work, and I would walk dogs for the independent living residents, earn a little bit of money, And then the afternoons, I would volunteer in the nursing home. And that's where I met, for the first time, people who were living with dementia. And it made a really strong impression on me. And so as early as middle school, I developed an interest in dementia and research, and really started thinking about how I could be a part of finding a treatment and hopefully a prevention for this devastating disease. And so just getting into some background about my research, we know that healthy lifestyle factors, so maintaining a healthy body weight, healthy blood pressure, healthy cholesterol, not having diabetes in early and midlife can decrease the risk of developing a dementia in later life, and also being physically active. And we know that in individuals who have some cognitive impairment, older adults with cognitive impairment, if they adopt multi-component interventions that include things like eating a healthy diet and engaging in physical exercise, that can actually slow down trajectory of their cognitive decline. And so if we could understand how diet and exercise are benefiting brain health, then perhaps we could use that understanding to develop treatments that could prevent or delay cognitive decline due to Alzheimer's disease and other progressive neurodegenerative diseases. In the past, and one of the things that's made this work possible is some really exciting developments in how we diagnose dementia and how we measure dementia pathology. So in the past, we had to basically get a sample of someone's brain tissue to definitively say that they had dementia due to Alzheimer's disease. We had to look for things like amyloid plaques and tau tangles that build up in the brain that are part of the Alzheimer's disease process. And so that really limited our ability to understand how things like diet and exercise can affect the development of dementia and Alzheimer's disease. Over the past 2 decades, we developed special imaging techniques that were able to measure the quantity of amyloid in people's brains and tau in people's brains. And also we developed a special lab tests that could measure the quantity of amyloid proteins and tau proteins in cerebrospinal fluid. And so that was a great advance that really helped push the field further. And also in the past few years, we've had even more exciting progress with the development of blood-based biomarkers that can be used to diagnose Alzheimer's disease. So we now have very sensitive and reliable blood-based biomarkers that can predict how much amyloid protein and how much tau protein are building up in the brain. One of these biomarker tests is called PrecivityAD, and it's actually available now in clinics. And then I expect that we'll have even better tests for Alzheimer's disease, a simple blood test in the next 2 to 3 years. And so not only will this help us with the diagnosis of cognitive impairment, but it will also tremendously help our research, because now we have very simple, non-invasive ways to measure how much amyloid and tau proteins people have in their brain with just a simple blood sample. And so with that development, we're now able to capture a lot of the different aspects of Alzheimer's disease. So this is a picture of a neuron in the brain and an astrocyte, which is a supportive cell that helps support brain health. And this is just a picture that describes the different biomarkers that we have that we can measure in blood that capture information about what's going on in the brain. So we have blood biomarkers that measure amyloid pathology, like amyloid beta 40 and 42. We have blood biomarkers that measure tau pathology, so p-tau or phosphorylated tau. And these two are very specific to Alzheimer's disease. So if patients have changes in these biomarkers, this is strongly consistent with Alzheimer's disease pathology in the brain. And then we have other biomarkers like total tau and neurofilament light, which measure neurodegeneration due to any cause. So it could be due to Alzheimer's disease, stroke, traumatic brain injury, other neurodegenerative diseases like frontotemporal dementia or Lewy body dementia. We also have other biomarkers like GFAP that measure neuroinflammation. And there's increasing evidence that inflammation plays a really important role in driving Alzheimer's disease and other causes of dementia. And with the development of these really exciting blood biomarkers, it's really allowed research to leap ahead to understand how things like exercise and diet affect brain health over long periods of time, from midlife when people are healthy and don't have any cognitive changes, all the way to the point in time where they develop a cognitive impairment and dementia. And so the goal of my study was to look to see if exercise training had a positive impact on these biomarkers in middle-aged adults who had cardiovascular risk factors like obesity, impaired insulin sensitivity, and lipid abnormalities or cholesterol abnormalities. And the idea behind this was to look to see if there is an exercise effect, what kind of exercise is having the greatest effect. Is it more about the amount of exercise or the intensity of exercise, aerobic exercise or resistance training? And if we found an effect, we could then look to see what other molecular changes are occurring that are responsible for this effect. And if we can identify these molecular changes, then we could develop drugs that target these molecular pathways that could essentially be like exercise in a pill. So you could get the beneficial effects of exercise for brain health in a pill, and that could potentially reduce the risk of developing dementia or help treat cognitive impairment in patients who already have dementia. And so in this exercise study, which was led by my mentor, Bill Krause at Duke and ECU, it recruited middle-aged participants, and they were all randomized to a 6-month structured exercise intervention. And so I quantified biomarkers in a subset of these participants at the baseline before they started the exercise program, and then after completion of the exercise program. Now, I encountered a lot of technical issues with the machine and also some delays due to COVID. So unfortunately, I don't have the full results to share with you yet. But I anticipate by the end of this year, we will have completed all the analyses and we'll have the results from the study available. But we did see some associations of these biomarkers that are associated with neurodegeneration and neuroinflammation, and saw that they were positively associated with age in this exercise study. And this suggests that as people get older, they have greater amounts of neurodegeneration and greater amounts of neuroinflammation. And so we hope to see if after completing 6 months of exercise training, if it can improve those or decrease the concentrations of those biomarkers, suggesting that exercise may be beneficially affecting neuroinflammation, for example. I also conducted a similar project in a study of caloric restriction, also in healthy young and middle-aged adults. And I found that in carriers of the ApoE4 allele, which is a genetic risk factor for Alzheimer's disease, caloric restriction was associated with a decrease in concentrations of a marker of amyloid production, suggesting that if people reduce their caloric intake, it may decrease the production of one of these amyloid proteins, and that might later in life translate to a decreased risk of developing dementia. And so the next steps for my study are to complete the planned assays. We plan to get the kits for the assays soon. They've been back ordered due to COVID, but hopefully by the end of the year, we'll have run all the assays and we'll be able to determine if there was an exercise effect on these biomarkers and what kind of exercise had the greatest effect. Is it aerobic training? Is it resistance training or weight training? And what type of aerobic exercise has the most potent effect? Is it high intensity aerobic exercise, or is it higher amounts of low intensity exercise? So these are all questions we expect to be able to answer over the next couple of months. once we've determined if exercise has an effect on these biomarkers, we're going to look at things like gene expression changes and also things like metabolites in the blood to see if we can identify the factors that are driving the benefits of exercise on these biomarkers. And then if we can develop drugs that target those factors, that may recreate the benefits of exercise on these biomarkers and essentially be like exercise in a pill for brain health. I'm also working on a career development proposal to do similar work in the caloric restriction study that I mentioned before, which is called Calorie. And ultimately, I hope to use this work to identify these drugs that recreate the effects of caloric restriction or exercise training and test them in older adults at high risk of developing cognitive impairment. And hopefully these drugs could become part of our kind of options for preventing cognitive impairment due to Alzheimer's disease, and also for improving cognitive function in people who have already developed Alzheimer's disease. And so I just want to thank my mentors, Bill Kraus, Kim Huffman, Janet Hubner, who helped with many of the assays, Carl Pieper, our statistician, and of course, the Dementia Alliance of North Carolina and all the wonderful donors who made this research possible. And I look forward to your questions at the end.
Thank you so much, Dr. Parker. I know we're going to have a lot of questions for you later, but we're really excited to learn more. And also, we may have to have you back to report back in at the end of the year or next year even to learn some more.
Happy to.
Great, thank you. Next up, we have Dr. Milta Little, and she is a doctor of osteopathic medicine. She's currently an assistant professor at Duke University's Department of Medicine and a geriatric medicine specialist with Duke Health. She received her DO from Midwestern University, Chicago College of Osteopathic Medicine, and did her internal medicine residency at St. Mary's Hospital Medical Center in St. Louis, and her geriatric fellowship at St. Louis University. Her funded research project, as you see here, was implementation of cognitive stimulation therapy across a healthcare system to improve clinical outcomes of individuals living with dementing illness. And we are so thankful to have Dr. Little with us today, and we're going to let you take it away.
Fabulous. Thank you all so much for this opportunity to talk to you about our exciting project. And as you saw from the title, this is an implementation research project specifically for cognitive stimulation therapy, which, if you are not familiar with this is a therapy program for people, designed for people with mild to moderate stage dementia of any subtype. And it is an in-person protocol that combines the ideas of orientation, reminiscence, and a Japanese learning technique called Saido, which really stimulates the brain to learn new concepts in new and interesting ways. And they combined these things into a 14-session protocol, which has been shown in previous randomized studies to improve cognition and quality of life for people living with dementia, at least as good as, if not better than, the current medications we have available. As I mentioned, this was designed to be an in-person group protocol of about 5 to 8 participants at a time. Thankfully, prior to 2020, it had been adapted to be able to be performed in a virtual format, although traditionally it is in person. But as you can imagine, we did utilize the virtual platform to a great extent for a majority of this project. So we had 2 broad aims that we wanted to cover. The first is really the model development of making the CST available here in our health system, as well as out into the greater community, by having a training program out of Duke that would then be able to disseminate out. And as I said, this was an implementation project. So we were going to be looking at a lot of measures of things, of process measures of how people were able to take what we were teaching them and implement it at their site. What barriers did they experience? What kinds of things helped them in this? And then really to kind of almost do a quality improvement process as we move forward through this project. Our second aim was to look at behavioral and psychological symptoms that can occur with dementia. This has been studied to some extent, but not to great extent. And so we were hoping to be able to start looking at the clinical impact of this as well. So just a very brief overview of our study design, as this is a slightly different type of project than the two that were presented, is that this is how we laid it out for things to be. So we were going to really engage with our community and form these collaborations, engaging sites, identifying people to be trained as facilitators, and then help them identify appropriate participants to go through the CST protocol. And then we would train the facilitators, help them in their site initiation with the materials and building that program, and then helping them over time to be able to maintain that and absorb that into their regular offerings. So we use the RE-AIM implementation framework, and we will be collecting a variety of different data, including some clinical data, demographic data, and a big piece of what we're looking at is qualitative data from a variety of different focus groups to really analyze how well are we able to take this protocol and disseminate it and implement it out and keep it to what it had been intended to do and really see the same impact on the outcomes for people living with dementia. So we did have a variety of adaptations that we had to do over the last year and a half. So our original model of training people to be trainers had to be suspended. Our in-person training from the CST Training Institute in St. Louis has been suspended until 2022. So, and also any in-person trainings for facilitators was suspended. So we had to get creative and work with our collaborators in St. Louis who have formed the CST, North American CST Training Institute. And we were able to negotiate virtual training sessions specifically for our facilitators in this area, as well as take advantage of some of their already scheduled training sessions. The CST delivery also had to be adapted. Initially, when we first started this project, nobody was doing group therapy at all. And specifically for our Duke University Health System, we had to convert everything to a virtual platform to be able to do that, which led to some delay because it takes time to adjust this into a virtual format. But there were a lot of opportunities that came from this as well, is that we were able to find some sites, namely the PACE programs, very quickly had to transition back to in-person because they really had to help their participants and bring them back, at least to some extent, back into their centers. And they were looking for things to be able to do to engage them and to help to prevent some of the decline that we were unfortunately seeing across the board in people with dementia over the last 2 years. And so we were able to find new sites to engage through this process. And we really hope to do a lot of this in person, including our focus groups. We wanted to really engage with people and build that community. But we did have to convert all in-person focus groups to virtual, which we did fairly quickly because people got used to this quickly. And we recorded audio only with permission. We're continuing to do that. So those were some of the adaptations we had to do. I know this is kind of a busy and an awkward table, but I'm going to spend the bulk of my time kind of focusing on this table because it can sometimes feel a little bit frustrating trying to navigate this, which is supposed to be an in-person protocol, and thinking we still want to do something that's meaningful for people. And I had a chance to look back and see what we've actually been able to accomplish. And we're pretty proud of ourselves for what we've been able to do to really get this protocol out to people. So when it came to our stakeholder engagements, what we had hoped to do and what we outlined for you initially was to engage 5 sites, 1 Duke University Health System site, and then 4 out in the community. And we had engaged some continuing care retirement communities or CCRCs and some senior centers in this as well. When those ultimately, when we started this grant, we're not doing anything in person and we're really focused on survival, we had to quickly revamp, as I said before. But because of that, we really reached out and made additional collaborations, and we now have engaged 8 sites. So we had 2 Duke University sites. Unfortunately, one disenrolled due to some competing interests. But we now have 5 community sites that have engaged with us. We have one Ccrc partner that, as it opened up visitation and activities again, are engaging in this process. One senior center, the Durham Center for Senior Life, DCSL, is now engaged. We have 3 PACE sites engaged, 2 fully trained and 1 hopefully trained soon. And then we were able to also engage with the Durham VA coach program, and they're going to be incorporating this into their offerings as well. And we have planned expansions still for the rest of this grant cycle, as well as in the future. We are hopeful to be able to expand across the state of North Carolina within the PACE programs. I was able to present to the state education PACE or education conference, and drummed up some excitement for this, and continuing to talk to people across the state. And even in South Carolina, I've had some interest. The Durham Center for Senior Life plans to open at their main site in Durham, and then later on expand to the second senior center site. And we are also exploring additional sites within our health system as well. So we have a lot of growth and excitement about this. In terms of the number of facilitators we would have wanted to train, we were expecting to train 2 at each engaged site. So we thought, well, we'll train 10 people by the end of this grant cycle. To date, we've actually trained 12. And on October 8th of next month, we will train an additional 12 to 16. I have, I think, 12 committed, and then a few others are still waiting to see how many we're going to be able to get on board. Participants, so people living with dementia actually participating in CST, we aim for 50 by the end of year 2. So far, we have 13 who have completed the protocol, either in person or virtually, or who will be completing within the next 1 to 2 weeks. We did have 6 dropouts. And with the expansion that we are seeing and the new people getting trained, we do expect to at least meet our target by the end of the 2 years of 50 participants. Our pilot group recruitments here at Duke, we had hoped to engage about a year ago, but as I told you, we had to do some adjustments with virtual, and we were able to finally start up our process here in the health system in August. BPSD, I'll talk about again a little bit later, the behavioral group. We had hoped to start in January, and that still is pending the in-person restriction lifting here at the health system. And then our focus groups, so kind of the meat of that implementation work. We didn't really specify. We weren't sure how many we would need to engage initially. So far, we've engaged 1 focus group with 4 CST facilitators. What we've done so far is immersed ourselves in the data. We have those transcribed, and we are working on our qualitative analysis currently, creating our plan and working towards analyzing those focus groups, as well as we have plans for at least 1 additional CST facilitator group, probably in January, once we get people trained and get their programs going, and then 2 CST participant focus groups, 1 virtual, 1 in person, once we have several groups that have completed their protocol, and we can engage them and get their input as well. So that's what we have done so far with our model development and implementation. And I have a variety of success stories that I also wanted to share with you. As I said, there have been, and as you may imagine, trying to do something designed in person, several points of frustration where I just felt like, I don't know that this is going to come together. But having such a strong team around me and really engaged and excited community partners have really led to some great stories that have been fed back to me. So within our pilot group, we had a lot of trepidation about doing this in a virtual platform. And can we engage people living with dementia in the same way and see similar outcomes? And what we found was that we did have to make some adjustments. So our facilitators here, instead of doing some of the traditional activities and games and get to know you things that we would do in person to build that community, they actually kind of flipped it and started with the conversations over current events and topics. And through that, they built trust with one another and really built a community and had great stories of how they've gotten to know one another. And one particular individual participant has really been instrumental in leading this group and bringing people together, and his excitement has just gelled all of them together. So we've been able to show that, yes, you can create community even through a virtual platform. Our Durham PACE folks are the ones that have really taken this on and have 2 groups that are finishing up right now. And I spoke to one of the facilitators a couple of weeks ago just to check in, see how things were going. And she very excitedly told me multiple stories. And my favorite one was baking banana bread. And for one of their group activities that week, they brought in ingredients, and the team, the group, the CST group, baked banana bread together. And through that process, they reminisced about when they used to do this in childhood, when they would bake with their children. A lot of stories emerged from that one activity. And then they also talked about, what else can we make from these ingredients? So the participants were able to look at what they had and say, oh, I could also make this. And so they were still learning new things and challenging themselves cognitively to take that next step, not just to talk about the past, but bring into the present and into the future, and to really engage them. And our focus group outcomes. So as I told you, we've immersed ourselves in the focus groups. We had some really great facilitators who gave some really great feedback that we were able to use almost on a quality improvement kind of path to take some immediate action steps to help to improve the success for our facilitators. So one opportunity that arose from that was the ability to take new facilitators and give them a chance to almost shadow some more seasoned facilitators and watch some virtual sessions. In addition, we have an opportunity to potentially beta test a virtual platform that was developed at the CST Training Institute specifically for CST. And because we're still going to be doing this virtually for, we don't know how long, we are toying with the idea of, can we also test this platform and see if this makes it easier for our facilitators and participants to engage. And then the state of Connecticut has a group of new facilitators who were also interested in developing a community of practice and have taken this on, and we have an opportunity to engage with them as well to really help our facilitators with this implementation. So as I stated before, with our behavioral group, we know that virtual is going to be a big barrier for them and a huge unknown variable that we're just not willing to wrestle with right now. And we all on our team prefer to wait until we can do this in person and really test it well. And so we will work towards that aim once we can resume that here at the health system, which is where it's going to be based from. And then next steps. So really what we're focused on is, how do we sustain this? We're not going to let this go at the end of this grant cycle, which we're so appreciative of this jumpstart. We're going to keep this going. So right now we're going to continue to recruit and analyze our focus groups. We're recruiting additional groups here, building capacity for both virtual and in-person delivery. We're going to continue to engage additional stakeholders. We have a variety of interprofessional students working here that we think would be great to engage in this to keep this going. I'm going to continue to work on this in the PACE program across the state. And through the Duke SNF Collaborative, we hope now that we're starting to get out of only survival mode in our Ccrcs and our nursing facilities and focusing on how can we engage the residents and do these activities, and we have some opportunities through there as well. We're going to be continuing our fidelity checks virtually, as well as the sites in person are doing their own internal, since we're not able to go in person to do those ourselves. And then we will be looking at the data. Hopefully very soon we'll start to get that rolling in. I think one thing that's important for you to understand is that we are not enrolling individual people living with dementia and consenting every single person to do CST. What we're doing is enrolling sites, and we are giving them this to then incorporate into their usual programming. And so their testing and their protocols are a part of their programming. They're collecting the data, they possess the data, they own it. Then they are going to fully de-identify, get rid of anything that would help us know or identify. If I walk by somebody in the street, I would have no idea whether or not they're in my study. And then we will get the data. So it's going to come just a little bit slower, but we have a data analysis plan once we do get that in. And then, of course, always eyeing the future funding opportunities, which we do have in line to be able to keep this going. We have some opportunities for that. And I know I'm a little bit over time, but I really want to take a few minutes to acknowledge the people that without whom I probably would have walked off the edge of a cliff, thinking that this was impossible. My mentor, Dr. Eleanor McConnell here at Duke, has been fabulous. She has extensive implementation science background and has been really instrumental in helping us get this off the ground. Of course, my project team, Dr. Kim Johnson, and my co-PI, who has been really instrumental in engaging sites, and particularly with the PACE engagement, and with our qualitative analysis, as well as our clinical research coordinator, Francis Caria, with really been working on with the focus groups, as well as helping us with our qualitative analysis. And then our facilitators and key personnel extraordinaire, Jennifer Sosensky and Ann Piopo. I have to mention them all specifically. And of course, the division support, my chief, Dr. Kalana Marique, Sue Doyle, who helped me with all the regulatory things, and Lynette Thacker, who makes sure that I am spending your money wisely, which we're so grateful for. And then, of course, the wonderful interprofessional team at the St. Louis University CST Training Institute. And with that, I will end. And thank you again so much for your support. And we now can open up for questions. I will stop sharing.
So much for that. That was wonderful. I'm going to turn this over to our gallery view here. I'm hoping. There we go. And if everyone wants to just unmute and jump in on these questions, we can do that. We've got quite a few questions in the chat. So I'm just going to go through and start with Dr. Ellis. We had a couple questions. One was to clarify about the knockout mice that you used in the study. Were the omega-3s taken away from those mice, or were they added to the mice?
They were taken away. So they had 50% lower. But we have been working in the lab to try to generate a model where we can go the other direction, where we can increase it, now knowing what we know. But we don't know yet whether or not that's going to work out as we would like it to. So data forthcoming.
Okay, well, we'll wait for the report on that. Another question would be, what type, would there be a specific type of omega-3s that you were involved? Was it general? And also, what might be the therapeutic level someone was going to look at if they wanted to increase their intake, their uptake of omega-3s?
Yeah, so the omega-3 that I study specifically is DHA, not ALA. So if you buy eggs or something that have omega-3s in them, ALA is usually what you're sold, but you can get milks and things that have DHA supplemented in them, which is what I study. So the DHA is the one that's really conferring the health benefits in the brain. There's another one called EPA, which seems to have better cardiovascular beneficial effects, but it simply is very low abundant in the brain. It's not there. And so it doesn't seem to be playing a huge role in the benefits. So DHA is the main one. And DHA is actually made by algae, and then fish eat algae, and then fish eat fish. And so you get it through fish consumption. Land plants make ALA. So that's another omega-3 that's, again, the one that's not necessarily super beneficial. So to get that special omega-3, you want to consume preferentially wild-caught fish that contain some fat in them. So if that helps to answer that question about the omega-3s, I would look for DHA. As for consumption, the recommendation is right now to try to have 1 to 2 servings of fish per week. So pretty achievable. There is data out there showing, I would imagine some people in the audience are familiar with the ApoE alleles. So ApoE4 versus ApoE3 and 2. ApoE4, has increased risk for Alzheimer's disease. But it's shown that ApoE4 carriers seem to be more responsive to omega-3 intake. They seem to respond well to that. And the thought is that ApoE4 allele was the major allele in individuals long time ago when fish was a major component of the human diet. And it wasn't until we sort of migrated and started alternative dieting that the other alleles sort of started emerging. So for example, so honestly, what I think is that there's probably a personalized nutrition level to this, which means you may have a genetic predisposition to be more or less responsive to different aspects of the diet. And so, my ultimate dream would be to someday try to sequence like our enzyme of interest, for example, and see how that correlates with omega-3 status of individuals. And so that you would know, oh, if you have some kind of a genetic change in this enzyme, then you need to, be really proactive with your omega-3 supplementation. But if you don't necessarily have ApoE4 allele or these other potentially genetic anomalies, then, maybe you won't benefit that much from the supplementation and just stick with, the one fish consumption per week. That's where I kind of see it going in the future, but we're simply not there. People have started doing this with a gene called FADS2, which also metabolizes omegas. It's not specific to the brain, but it's so people are getting themselves sequenced to find these genetic changes and then using that information to know how to change their diet. So I think that we might be moving in that direction. So that's my dream, to be able to sequence ACSL6.
Well, we wish you luck with that. We hope you're able to succeed in that. Thank you. Dr. Parker, we have a couple of questions for you. And one is, so the biomarker test or screen that you mentioned that's available now, is that available to all providers or just at specialty clinics or memory clinics? And a part 2 of that, is that done through spinal fluid?
So I think the test you're mentioning or interested in it's called the Precivity AD test. And it's pretty new. So there are a lot of clinics and providers who aren't familiar with it. The major issue is that it's not yet covered by insurance, so it is out of pocket, and it costs about $1,250 for the test. So when I talk to patients about it, I offer it as an alternative to the spinal tap because it is a blood-based test. So if people are, hesitant about a spinal tap, then, that is an alternative. But I try to encourage patients to go for the spinal tap or the lumbar puncture because it's a much better test and it's a much more sensitive test and gives much more of a clear-cut answer. But for those patients who are hesitant to undergo a lumbar puncture, have some contraindication to a lumbar puncture, the Precivity AD test is available. And if you just Google Precivity AD, it will come right up. And it's just the major hurdle is the cost at this point and the fact that it's not yet covered by insurance.
Okay, thank you. Let's see, let's jump over to Dr. Little for just a moment. First of all, congratulations on all of the transitions that you made during COVID. you didn't pull all your hair out, and it looks like you're making amazing progress. So congratulations on that. I don't know if you can answer this question, but there was a question about which CCRC is participating.
Yeah, I think it's fine to, I called out some of the others. So it's the Forest at Duke is the current participating CCRC. We certainly hope to expand to additional ones in our area soon.
Great. I think we might have a couple people on from the Forest at Duke today, so that's nice. Can you give us a few more specifics on CST itself, kind of how that works? And then as part of that, maybe what is most important for your facilitators as they're working with the people living with dementia for them to notice or keep in mind while they're doing those things?
Yeah, those are great questions. So CST is, as I said, it's a small group protocol, usually between 5 to 8 participants, and it's a 14 session protocol that's laid out. I should have had a booklet with me. I mean, I have them, I just can't reach them, but I've run out of the camera view. But there are a variety of booklets that go through how to run a session. They're 45 minutes to an hour long, and they run through, the thing about them is that you run it the same way. So every session opens the same way. There's a welcome, an activity, an article, and then a closing, and there's orientation parts in there. So, but every session has a different kind of theme per se. So one day it might be a cooking theme, the next time it might be a shopping theme, just kind of throwing things off, but the same kind of process for each one. So there's familiarity, and then each one builds upon the other as well. So trying to kind of move these concepts through. And then after that, there's maintenance. So hopefully we can keep people engaged, and then it's twice a week for 14 sessions. And then after that, it's weekly maintenance after that. The facilitators don't have to have any special training other than getting CST. We do say that it's best if it's people who understand how to work with people living with dementia and who have experience doing groups, because their role really is working through these sessions. One person kind of monitors to make sure everybody stays engaged while the other one runs the different activities. Medicare will reimburse for certain special professions. So occupational therapists, speech therapists, and social workers, with a variety of different ways that they bill, can bill under Medicare and other insurances for running these groups. So that's one mechanism for sustaining this. But anybody can be trained to be a facilitator. We have multiple recreational therapists, because it also really falls in their purview as well, who get trained for this. So hopefully that answered the question.
Thank you. We did have one question from Patty. It's a rather long question. I don't know if you all were able to read it in the chat. I will try and give you the gist of it. But she said she had an elderly aunt who, after a fall, needed a subdural hematoma evacuated, and then afterwards she began having illusions and delusions that were kind of out there about aliens and eating babies and all kinds of things. She contacted the police, wrote "help me" on her window, and she's wondering, and I guess this is for you who are practitioners perhaps, and I have an idea of the answer, but would you attribute this more to brain injury, fall, surgery, or dementia? And I know there's not a lot of information there, and we can't diagnose somebody from this, but I guess we could maybe expand this a little bit and say, when you do tend to see a lot of delusions in people that you're treating, do you tend to think that that's dementia perhaps, or something else? And I'll leave that open to everyone.
Elta, do you want to go? Or?
Yeah, I can. You were unmuted, so I didn't know if you had it.
No, I think we'll probably say the same thing. So
probably. I mean, I certainly any dementia subtype can be associated with hallucinations and delusions. Certainly some are more commonly associated with than others, such as dementia of Lewy body. However, for that to have happened so suddenly that way, to me, says that probably more related to the traumatic brain injury. And certainly that's something that I have personally seen in my practice, as well as what you see described. It's kind of the sudden behavior changes after trauma. I don't know if you have anything else to add, Daniel.
Yeah, so, I think dementia, and then certainly after traumatic brain injury, and then also, delirium can also be associated with delusions and hallucinations. And we see that in older adults who've been exposed to some kind of medical stressor, whether that's a fall or a head injury. And so my guess would be, without actually evaluating the patient, probably a combination of the three.
Okay, thank you very much. I also wondered if any of you had questions for each other. I know Dr. Little and Dr. Parker, you talk a lot, so you may not, but anybody have any questions for each other? Okay, well, I think we're going to go ahead and end this because I know we have some time constraints, but on behalf of Dementia Alliance, we want to thank all of you for joining us, for sharing your findings, for being so dedicated to all of your areas of practice. We look so forward to following what you're doing and hearing back. So even after you're done reporting with this, just know we'll be watching, and we look forward to hearing more information about what you're doing and cheering you on, as I know all of our families and participants have. So thank you all so much for that. I also wanted to thank the members of our scientific advisory panel for helping us to find these wonderful researchers, and to Laura Gaddis, who joined us earlier, and Guardian Angel Thrift for helping us make this a possibility, to all of our donors who have given toward research, and to our corporate sponsors, Aware Senior Care, Capital Financial Solutions, and ESI for helping us make all of our virtual programs possible. So thank you all for joining us today. Best of luck in your future endeavors, and we really appreciate you being here. Everybody have a great day.